Hi everybody, this is a continuation of the DNA replication lecture. In the last segment we were talking about DNA polymerase and how a DNA polymerase functions. Now I'm going to go through an animation of a typical DNA polymerase so that so you guys can see how the DNA polymerase activity and the exonuclease activity of a typical DNA polymerase interplay. So here's the schematic of a DNA polymerase enzyme, obviously protein. Here are the fingers that bind the nucleotides. Here's the thumb of the DNA polymerase that closes on the enzyme when the enzyme is working in a processive fashion. It is the palm of the DNA polymerase. It contains the active sites for the polymerase reaction. So two polymerase active sites. There's two spaces for the two nucleotides that are being added, and then the other one is in wait. And as well, the very important exonuclease site sort of towards the thumb of the palm area. So, in this particular animation, we're going to rotate the DNA polymerase. So it's a little bit easier to see how things go. And this is how your DNA polymerase is represented in your book as well in all the different diagrams. So now here's the DNA. The blue strand is a chunk of the template DNA. Just assume that it continues going here. And I only draw it partially for ease. The red DNA strand is the synthesized strand that the DNA polymerase has already made. And so the DNA polymerase will be moving towards the right on this slide. So what happens when the polymerase made a mistake and mispaired deoxycytidine against the thymidine instead of an adenine? So there's our cytosine and has been incorporated. The nucleotide has been incorporated, the phosphates have left. There's the DNA polymerase synthesizing the phosphate bond. It doesn't actually wiggle, but it's a nice way to animate enzymatic activity. And then there we go, it's attached to the chain. And then when the DNA polymerase realizes that there's incorrect base pairing going on, the C is bulging out and not forming the proper base pairing with the thymidine, like an adenine would. So what DNA polymerase will do next is to reposition the mispaired, sorry, it shouldn't be saying misfired. My keynote constantly fixes things without me realizing them for me. So polymerase repositions the mispaired DNA C terminus here into the 3 to 5' exonuclease site, so right here. See how it has moved itself on the DNA and repositioned so that the DNA is now within the 3 to 5' exonuclease site, which, because only that site is able to efficiently catalyze the removal of a nucleotide reaction, so the exonuclease reaction. So the exonuclease in the active site of the exonuclease active site, the exonucleotide removal reaction occurs. So the connection between the G and the C, the phosphodiester bond right here, is going to be hydrolyzed. There we go, hydrolysis is also wiggle. And then now the C is no longer attached to the freshly synthesized DNA strand and it diffuses away. And then, now that it has removed the incorrectly base paired nucleotide, the DNA polymerase will then proceed to try to add a correct new one. So the 3' DNA terminus is now going to be repositioned again back to the polymerase site of the DNA polymerase where the polymerase reaction can be catalyzed. There we go. And the polymerase will now attempt to incorporate another nucleotide, hopefully a correct one, the adenine, right? So there comes an adenine. It's base paired with the T, and the DNA polymerase catalyzes the phosphodiester bond formation between the growing DNA chain and the freshly added nucleotide. And this is the diagram from your book, which was basically what this animation was. So you guys can go over it again. It's the diagram 11.6 in your book. Now, we have just talked a lot about how DNA polymerase works, but the very important realization about DNA replication is that DNA polymerase does not do it alone. DNA replication requires a coordinated effort between 9 or 10 different enzymes, which together, because they're all located in the same space, are called the replisome. So if you see, this is just the very basic diagram of how DNA replication fork functions. And here you don't see any of the enzymes. It's only the blue template DNA strand and the red growing synthesized strand, as well as the Okazaki fragments with their RNA primers that are depicted. And here's the list of the proteins that participate in a eukaryotic replisome. You guys don't need to know the names of the proteins, but you need to know their function. That there's different proteins are fulfilling different functions, and all of these functions are important. So if you guys remember from the beginning of this lecture, the ORC complex is the initiator of DNA replication. There's helicase loaders that need to recruit the helicase to the ORC region. The MCM27 is a helicase, etc., etc., etc. You guys can go through the diagram on your own. And so the previous slide was showing you the laundry list of all the enzymes involved, all the proteins involved. Here's how the proteins are actually positioned in the three-dimensional space. Because most of the proteins in the DNA replisome are actually tightly associated. The Okazaki fragment that's in the previous diagram were located here, and here are actually located pretty close to the second, the leading strand DNA polymerase. So as a result, because the lagging strand has been bent, so you guys can see it's bending here. This is the lagging strand of the DNA of the DNA replication. This is the leading strand. It has to be protected, and it has to be the single-stranded area of this DNA, of the template DNA, which is in orange, has to be protected. So let's talk about all the different players. So first of all, you need a DNA helicase. In fact, when you initiate DNA replication, that's one of the first enzymes to be recruited to the origin of replication sites inside DNA. It works really fast. It unwinds DNA at 10,000 rotations per minute. And it produces two single-stranded DNA strands. One of them is the leading strand right here, and then the lagging strand right there. Next comes the primase. Specifically, there was a primase here originally to initiate replication on the leading strand as well, but after that, the DNA polymerases of the leading strand has taken over and no longer needs another primase in there, because on the leading strand, the DNA can just happily synthesize DNA at in the 5' to 3' orientation. But you always need a primase constantly goes on and off on the lagging strand to synthesize the little RNA primers, which will be extended into Okazaki fragments by the lagging strand DNA polymerase. I'll talk about the beta clamp. Yeah, let's talk about the beta clamp for a second. So the beta clamp, which are the red ring here and this red ring right there, are very important both in eukaryotic and prokaryotic DNA replication. It's called the beta clamp in prokaryotes and PCNA in eukaryotes. And it's critical because that's what actually positions the DNA polymerase onto the DNA. Because the DNA polymerase's association with the DNA molecule is not that tight. It is the clamp that holds it there. So The clamp holding the DNA polymerase onto the leading strand just stays with the DNA polymerase. However, since on the lagging strand, the DNA polymerase constantly has to come on and off, as soon as it finishes the Okazaki fragment, it will come off, and then will be recruited again to the next RNA primer location. The pCNA is the first thing that will bind the RNA primer once the primase has finished synthesizing it. And it drags the DNA polymerase along with it. DNA polymerase is a much bigger critter. And then this text right here and these brown guys right here are single-strand binding proteins, single-strand DNA binding proteins. They code the lagging strand until the primase can get there and synthesize a primer, and the DNA polymerase can extend the new strand. The reason the single-stranded DNA has to be coded is because the helicase just did this, important job of unwinding, breaking the hydrogen bonds between the base pairs. If single-stranded DNA is allowed to float by itself, it will assume some sort of secondary configuration, preventing the primase and the DNA polymerase from easily accessing the DNA and allowing it to serve as a template. As a result, positively charged single-strand DNA binding proteins base pair with a single-stranded DNA and keep it from folding. So that's the basic. These are the basic members of a typical DNA replisome, both for eukaryotes and prokaryotes. A helicase, 2 polymerases, 2 beta clamps, two clamps, pCNA clamps that are holding the polymerases to the DNA, primase to synthesize primers for the lagging strand, and single-strand binding proteins to code the single-stranded template of the DNA to prevent it from having secondary structure. Now, another important protein that's essential for DNA replication but is not necessarily part of the actual replisome is the topoisomerase. Topoisomerase is a protein that makes small nicks in the DNA by cutting a phosphodiester bond once in a while. The reason you need a topoisomerase is because when the helicase is pushing the double helix apart and it moves really fast, it creates torsional stress further down the DNA molecule. So showing here, if you have two tightly wound strings, which is what DNA strands can be thought of as, and you are unfolding, you're ripping apart, you're unwinding the DNA in this end, the torsional stress that's created will create supercoils inside the DNA or inside of your little rope that will create these major roadblocks for the motion of the replisome, because now it's triple wound. As a result, the idea for topoisomerase is that it will nick DNA here and prevent these supercoils from happening. So let's look very briefly at how topoisomerase works. You guys can see again that here's the helicase moving along the DNA and unwinding it. And because it's unwinding it in this area, the DNA is becoming sort of more tightly wound in this area. However, and the end result of this would be the supercoil forming. You can try this at home. Just take 2 pieces of string, wind it, and see that if you have wound the strings and attach them to some sort of solid support like a chair, and then begin to unwind the 2 strands, you will get these knots, these supercoils forming in the strings. The topoisomerase will come and make these little single-strand breaks. It does not change the base pairing of the 2 DNA helixes. It does not remove any nucleotides. It just breaks phosphodiester bonds periodically. And because it breaks the phosphodiester bonds periodically, it relieves the torsional strain that's caused by the helicase. Okay, now back to the trombone model of DNA replication. The reason it's actually called a trombone, because it kind of looks like a trombone. There's this extra lagging strand loop, like the trombone that has this big loop that you move back and forth when you play it. So this is a figure from your text. You can study that, and you can. But instead of, I wanted to take advantage of the fact that we are all sitting on computers here. So you guys, so instead of going over this diagram, I recommend watching this video. So I'm going to pause my recording and get to the video for a second here. All right. Oh, see, I tried to do it smoothly and didn't work. Here we go. So this is a DNA replication video from HHMI, which is an important research institute. So there's some talking towards the end of the video, but here I'm going to be pausing it and just pointing out the players as well. So this blue guy right here, so this is the unreplicated DNA coming in. This blue guy right here is the helicase. You can sort of see its hexameric structure. If I play the video, you can see how it's rotating really rapidly and unwinding the DNA. And because it's unwinding the DNA, it's producing these two single-stranded DNA molecules, right? So this is the leading strand right here, and it's promptly being, a new DNA is promptly being synthesized right here. And this is the lagging strand, and just imagine it's going out of your screen and coming right back to be replicated right here. There we go. Do we see it? Right there. And this is a lagging strand being replicated. You can see that there's two clamps, the purple clamps that are holding the green DNA polymerases right there. And there's, in this case, two polymerases sitting on the lagging strand synthesizing the DNA from the two different RNA primers. So now we have zoomed out, and you can again see the trombone model. In this case, the loop is not as obvious here. This is the leading strand, and this is a lagging strand, and this is the big clamp. Eukaryotes have a bigger clamp holding the, in this case, two polymerases together, and one polymerase just fell off. There we go. Did you guys see how this polymerase has now attached itself to the single-stranded DNA? And they have removed the RNA binder, the DNA binding proteins from here, just so you can see the structures better. There we go. There it goes. It's going to begin to synthesize now. There we go. Did you guys see how it's synthesized? All right. So I'm going to let you guys watch the video on your own. I recommend watching it a few times because the video moves pretty fast, so that you guys can appreciate the many players and of the DNA replication and how these proteins interact with each other. Okay, now let's wrap this up because I have only a few slides left. So most of your book and your previous knowledge probably bases itself on prokaryotic replosome. Here the players are very well known. There's the helicase, there's the primase, there's the polymerases with the beta sliding clamp that holds the polymerase onto the DNA. And then there's the big clamp loader that coordinates the loading of these three polymerases. And this bump out here is the trombone version. In this case, there's two functioning. These blue little guys are single-strand binding proteins. And if you compare it to the image that you also have of your book of how putatively the eukaryotic replosome works, in this case from yeast, you see how very similar these two structures are. Again, there's a helicase, there's a primase, there's a clamp, in this case called pCNA, and as well as polymerase, the polymerases, epsilon for the leading strand and delta for the lagging strand, and as well as there is a clamp that loads the clamp loader that will take the clamp and the polymerases on and off. Here's the comparison of the different players. Again, a helicase, both eukaryotic and prokaryotic replisomes have one. Everybody has a primase. In case of polymerases, the eukaryotes have two different polymerases. one for the leading and one for the lagging strand, while the prokaryotes have one and the same polymerase. But in both cases, there is a clamp complex, clamp loader complex, single-strand DNA binding proteins, and enzymes that play a role in Okazaki primer repair by removing the RNA primer and filling it in with DNA. And of course, you need a ligase to fuse the Okazaki fragments together on the lagging strand. And as I said before, and I'll say it again, you don't need to know these names, but you can if you're curious, if you like it. But you do need to know their functions. And like the names of the proteins, the general names of the protein, what functions they fulfill. This is something I found on Pinterest and I thought it was funny. Now,